Introduction. We have discussed structure of eukaryotic cells and structure and functions of cell membrane and cell wall already. Yes, now we will discuss structure of endomembrane systems. Okay, we will also observe structure of endoplasmic reticulum, Golgi apparatus, lysosomes, vacuoles, mitochondria, plastids, and ribosomes.
Objectives. At the end of this lesson, you'll be able to find the characters of endomembrane systems, find the characters of endoplasmic reticulum, understand characters of Golgi apparatus, find the characters of lysosomes, find the characters of vacuoles, find the characters of mitochondria, understand characters of plastids, find the characters of ribosomes. Characters of endomembrane system. A system of interconnected membranes, also known as the cytomembrane system, that fills the cell interior and connects the cell boundary with the double-membraned organelles, nucleus, plastids, and mitochondria. The phospholipid bilayer forms the basis for both outer and internal membranes. Thus, while the plasma membrane, the outermost cell membrane, is an integral component of the cell boundary, its numerous convoluted protrusions into the cell interior and its connectivity with the cytomembranes also allow it to be considered a part of the endomembrane system. The endomembrane system organizes the eukaryotic cell into numerous specialized functional domains that are recognized as different organelles. Major endomembrane-derived organelles of the plant cell are endoplasmic reticulum, Golgi bodies, peroxisomes, vacuoles, endosomes, autophagosomes, outer nuclear envelope. Structure of the endoplasmic reticulum. The endoplasmic reticulum ER is an organelle of cells in eukaryotic organisms that forms an interconnected network of tubules, vesicles, and cisternae. The lacy membranes of the endoplasmic reticulum were first seen by Keith R. Porter, Albert Claude, and Ernest F. Fulham in 1945. There are two regions of the ER that differ in both structure and function. One region is called rough ER because it has ribosomes attached to the cytoplasmic side of the membrane. The rough ER is a series of flattened sacs. Rough endoplasmic reticula synthesize proteins. The other region is called smooth ER because it lacks attached ribosomes. The smooth ER is a tubule network. Smooth endoplasmic reticula synthesize lipids, steroids, and morphine, metabolize carbohydrates and steroids, and regulate calcium concentration, drug metabolism, and attachment of receptors on cell membrane proteins. The space inside of the ER is called the lumen. The ER is very extensive, extending from the cell membrane through the cytoplasm and forming a continuous connection with the nuclear envelope. Characters of Golgi apparatus. Golgi apparatus is a membranous organelle composed of many flattened sacs. The Golgi apparatus forms a part of the cellular endomembrane system. It was identified in 1898 by the Italian physician Camillo Golgi, after whom the Golgi apparatus is named. The secretions of cells are packed in them and transported from the cell. Golgi apparatus consists of a set of membrane-bound cisternae, vesicles, and secretory vesicles having polarity. It processes and packages proteins inside the cell, and before they make their way to their destination, it is particularly important in the processing of proteins for secretion. Characters of lysosomes. Lysosomes were discovered by the Belgian cytologist Christian D. Dove in the 1960s. The name lysosomes derives from the Greek words lysis, to separate, and soma, body. They are frequently nicknamed suicide bags or suicide sacs by cell biologists due to their autolysis. Lysosomes are cellular organelles that contain acid hydrolase enzymes to break down waste materials and cellular debris. They can be described as the stomach of the cell. They are found in animal cells, while in yeast and plants, the same roles are performed by lytic vacuoles. Lysosomes digest excess or worn-out organelles, food particles, and engulf viruses and bacteria. The membrane around lysosomes allows the digestive enzymes to work at the 4.5 pH they require. They are created by the addition of hydrolytic enzymes to early endosomes from the Golgi apparatus. Lysosomes fuse with vacuoles and dispense their enzymes into the vacuoles, digesting their contents. Characters of vacuoles. A vacuole is a membrane-bound organelle which is present in all plant and fungal cells and some protist, animal, and bacterial cells. Vacuoles are essentially enclosed compartments which are filled with water containing inorganic and organic molecules, including enzymes in solution, though in certain cases they may contain solids which have been engulfed. Vacuoles are formed by the fusion of multiple membrane vesicles and are effectively just larger forms of these. They have no basic shape or size. Its structure varies according to the needs of the cell. The functions of the vacuole includes isolating materials that might be harmful or a threat to the cell, containing waste products, containing water in plant cells, maintaining internal hydrostatic pressure or turgor within the cell, maintaining an acidic internal pH, containing small molecules, exporting unwanted substances from the cell, allows plants to support structures such as leaves and flowers due to the pressure of the central vacuole. In seeds, stored proteins needed for germination are kept in protein bodies which are modified vacuoles. Characters of mitochondria. Mitochondria are membrane-bound organelles and, like the nucleus, have a double membrane. They are about the size of bacteria but may have different shapes depending on the cell type. The outer membrane is fairly smooth, but the inner membrane is highly convoluted, forming folds called cristae. The cristae greatly increase the inner membrane's surface area. It is on these cristae that food, sugar is combined with oxygen to produce ATP, the primary energy source for the cell. Mitochondria has its own DNA and ribosomes. Mitochondria provide the energy a cell needs to move, divide, produce secretory products, contract. In short, they are the power centers of the cell. Characters of ribosomes. George Palade in 1953 under electron microscope observed dense particles. Ribosomes are granular structure. Ribosomes are made of ribonucleic acid RNA and proteins. They are not membrane bound. The eukaryotic ribosomes are 80S and prokaryotic ribosomes are 70S. S is Swedberg unit and stands for the sedimentation coefficient. It is a measure of density and size. Both 80S and 70S ribosomes are made up of two subunits. Ribosomes are also attached to the outer membrane of endoplasmic reticulum. Ribosomes take part in protein synthesis. Characters of plastids. Plastids are found in all plant cells and also in euglenoids. They are large in size, so they can be easily observed under the microscope. They have some specific pigments, so they give color to the plants. On the basis of pigments present in plastids, they can be of three types: chloroplasts, chromoplasts, and leucoplasts. The chloroplasts have chlorophyll and carotenoid pigments which are responsible for trapping light energy essential for photosynthesis. Chromoplasts have fat-soluble carotenoid pigments like keratin, xanthophylls. They give yellow, orange, or red color to the plants. Leucoplasts are colorless plastids of various shapes and sizes. They are amyloplasts which store carbohydrates, example, potato, elaioplasts which store oils and fats, and aleuroplasts which store protein. Characters of chloroplasts. Chloroplasts are found in mesophyll cells of leaves. They are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles ranging from 5 to 10 micrometer long and 2 to 4 micrometer wide. There may be one chloroplast, as in Chlamydomonas, or 20 to 40 per cell, as in green algae. Chloroplasts are double membrane bound. The inner membrane is less permeable. The space limited by inner membrane is known as stroma. There are a number of organized flattened sacs called thylakoids in stroma. These thylakoids are arranged in stacks like the piles of coins and are known as grana. The thylakoids of different grana are connected by the flat membranous tubules called stroma lamella. The space enclosed by membrane of thylakoids is known as lumen. The stroma contains carbohydrates, proteins, double-stranded circular DNA, and ribosome 70S. Thylakoids contains chlorophyll pigments as well. Did you know? Lysosomes were earlier known as pericanalicular dense bodies. Main enzyme present in Golgi apparatus membrane is nucleoside diphosphate. Liver cells have both smooth and rough endoplasmic reticulum. Mitochondria and chloroplasts are self-duplicating cell organelles. In giant amoeba chaos, there are about 50,000 mitochondria. Summary. Let us summarize what we have learned. SER is mainly found in cells actively engaged in lipid metabolism. The smooth endoplasmic reticulum SER is detoxification site in liver. Organelles connected with lipid synthesis are SER. Granular endoplasmic reticulum differs from SER in having ribosomes on its surface. Gastric cells secreting zymogen have well-developed RER. RER is well-developed in cells engaged in synthesis of secretory products. Function of RER is protein synthesis. RER differs from SER in presence of ribosomes. The Golgi complex occurs in all cells except erythrocytes and bacterial cells in perinuclear area. They are absent in bacteria and blue-green algae. The Golgi complex is a membranous organelle composed of flattened sacs. The secretions of cells are packed in them and transported from the cell. The Golgi complex is specialized for glycidation of lipids and proteins. Golgi complex produces vesicles and lysosomes. ETC is component of Golgi complex. Golgisome is involved in transformation of cell membranes and protein packaging. The Golgi complex is exported house of the cell. Lysosomes were discovered by D. Dove. They are called so because they have carboxylating enzymes. Lysosomes are formed Golgi bodies. Lysosomes are also called suicidal bags because they have hydrolytic enzymes which function at acidic pH 5. Lysosomes are single membrane structures containing enzymes for digestion of all types of macromolecules. Double membrane or unit membrane is absent in lysosomes. Lysosomes are membranous bag with hydrolytic enzymes which is used for controlling intracellular digestion of macromolecules. Lysosomes are polymorphic and contain hydrolases. Lysosomes reduce the number of other organelles. Autolysis is connected with lysosomes. If lysosomes get ruptured in a cell, it dies. Function of lysosomes is breakdown of cell substances. When lysosomes fuses with phagosome, it results in the formation of secondary lysosomes. Cell vacuoles contains water and dissolved substances. Tonoplast, a differentially permeable membrane, surrounds vacuole. Vacuoles are of four types: contractile vacuoles, sap vacuoles, food vacuoles, and gas vacuoles. Vacuoles store food and waste. Some vacuoles store extra water. Cell sap occurs in vacuole. Function of vacuole is storage, waste disposal, and cell elongation and protection.